Okay, we have gone through all the videos for gene expression or protein synthesis, but there's one other topic that I want to talk about that people don't think about that much, or it doesn't come into their thinking when we're looking at passing on chromosomes during cell division, mitosis and meiosis that we looked at, and passing on genes to the next generation when you reproduce, or inheritance, heredity. And that is this idea of gene regulation. So if you think about two cells in your body, a skin cell and a nerve cell, both of those cells have the same DNA, but they do different things. They're different structurally, they're different functionally. So how can those two cells do two different things and look two different ways and have the same exact DNA? Well, that's what we're talking about in terms of gene regulation. And we're going to look at a number of different aspects of that. It's not going to take very long, but it's an interesting topic to think about. So we're going to go over some basic concepts and genes making RNA. It's something else that genes are used for, not just making proteins. And then how these genes are regulated and some of the different circumstances under which some of the DNA is expressed and used to make proteins and some of it isn't. So first of all, what is gene expression? Gene expression, as we've been looking at it, is taking a gene within DNA, using that to make RNA, and using that RNA to make a protein. That's what we saw in transcription and translation. That's what happens at the ribosomes in the case of translation, and happens on DNA at the site of DNA in terms of transcription, where you're making RNA. Well, if you look at prokaryotic cells, bacteria, basically, they have only got one chromosome. They don't have that many genes. They don't have that much DNA. And most of that DNA is involved in making proteins. And most of those proteins have to do with energetics. Bacteria just using a certain number of enzymes to get a certain amount of energy to power its little tiny cell. If you look in our cells and other eukaryotic cells, we've got a lot more chromosomes, a lot more DNA, a lot more genes. We've got these 2.9 billion pairs of nucleotides, as many as 25,000 genes. That's a lot of different parts of DNA that can be used to make a lot of different proteins. So if you look at our DNA, there's DNA that's used for other things besides making proteins. We've got a lot of non-coding parts of DNA. Some of that we're going to look at in a minute is used to make RNA, not proteins, but RNA. And then some of that isn't used to do anything. It's very repetitive, it's very conservative. My DNA is going to look like your DNA, it's going to look like everybody's DNA, because it's not used to make proteins. It's not really a part of the DNA that serves as a gene. It's more of a structural DNA. So if you look at the genes that we have, humans, our genes, what do they do? What do these genes do? Well, a lot of them are used to make proteins, as I said. If you look at this pie chart here, there's this type of protein that breaks things down, and there's this type of proteins move things, this type of protein that's involved in nerve transmission, this type of protein breaking things down, and this carbohydrate binding transport protein and protein binding transport. Look at all these different enzymes that are involved. There's electron transport and transcription and transport, all these different functions of proteins, a lot of which are enzymes. And then you have some other things. It's membrane proteins that do different things, and cell adhesion. There's a whole bunch of different functions for proteins that are made based on information that we have in our genes that are relayed to ribosomes through RNA, and then those proteins are made, and then they carry out these different functions. So there's a whole bunch of proteins that we use for a whole bunch of different things, many of which are enzymes. Well, if you look on one particular chromosome, for instance, chromosome number 1 of humans, there's 246 million base pairs. That means on one side of the chromosome, the DNA in the chromosome, there's a sugar and a phosphate and base. And on the others, there's a corresponding base, a complementary base, and another sugar and phosphate. So 246 million of those just on chromosome number 1. Look at chromosome number 7, 158 million pairs. And if you looked at chromosome number 1, now we've mapped out the entire human genome, the Human Genome Project. So we know on all 23 of our chromosomes where these different genes are, what the DNA sequence is, or the sequence of those bases. Here's all these things. You don't have to zoom in on this or even be concerned about what all these words are, but there's all these different genes on chromosome number 1 that synthesize all these different proteins, have all these different functions. Same thing on chromosome number 7. All these different genes on chromosome number 7 are involved in making all these different proteins. So that's a lot of what our DNA does, and that's a lot of what our genes do. Let's look at another aspect, very briefly, and not in very much detail, but another thing that genes do is not make proteins, but they make RNA. RNA, as you've just seen, has an important role in making proteins. You need RNA to be made, and you need RNA to carry the information from DNA to ribosomes to make a protein. So you've got a number of different proteins that are made based on genes, and then you've got this the need for RNA. So there's a number of genes that are involved in the synthesis of RNA. And there's a number of there's these RNA polymerases. Each one of these RNA polymerases is a protein. It's an enzyme that makes RNA. There's different types of RNA that are made in different parts of this is in the nucleolus, which is part of the nucleus. And this is it's not the cytoplasm of the nucleus, but it's a fluid outside of the nucleolus where these different RNAs are synthesized. So you know we've got 3 different types of RNA, messenger RNA and transfer RNA and ribosomal RNA. They're all made in different parts of the nucleus by different enzymes and based on different genes in our DNA. So it's not just proteins that we're making that are going to be enzymes that are breaking down our food or something like that. There's a number of other things that are made. These enzymes are used to do a whole bunch of different things related to a whole bunch of other macromolecules. But one of the main things that genes do, besides make proteins, is they make these proteins that are specifically geared towards making RNA. Because the RNA is very important, and we need a lot of RNA. All right, well, here's the main thing that I want to talk about today. And as I said, this won't take that long to go through, but remember this DNA, you get DNA in your chromosomes, you inherit that from some other cell, whether it's one of your parents or your own cells are dividing and making new cells. So a cell has a certain amount of DNA. Within that DNA, there's a whole bunch of genes. Some of that DNA is not used to do anything. So that's non-coding DNA. It's not used to make a protein. It's not used to make RNA. And remember, when we were looking at inheritance, there were these dominant alleles and recessive alleles. And remember, another thing that you realize is that these chromosomes come in pairs, homologous pairs. These traits, a lot of them come in pairs. So you've got 2 possibilities. For instance, for tongue rolling, you could have a gene on one of your chromosomes that results in proteins being synthesized that are related to the muscles in your tongue that allow you to roll your tongue. If you've got one dominant allele, you make that protein and you can roll your tongue. But remember, you've got 2 chromosomes. whichever chromosome this is on. Let's say that the tongue rolling proteins are all on chromosome number 6. You've got 2 chromosome number 6s. So as long as you get one with the version of a gene that makes the protein that allows you to roll your tongue, you're going to be able to roll your tongue. On this other version, maybe it's your mother's version of chromosome number 6, you could have the recessive allele. And so if that gene was ever expressed, it would result in a protein that's not going to allow you to roll your tongue. If you've got 2 of those recessive alleles, you don't have a dominant allele, then there's no way for you to make the protein that allows you to roll your tongue. All you're doing is making the proteins that are part of your tongue muscles that you're unable to roll your tongue. But if you have the dominant allele and the recessive allele, one on your father's chromosome number 6 and one on your mother's chromosome number 6, then they're both not expressed. They're both not used to make proteins. Only the dominant one is. That recessive allele, that gene, sits in your DNA your entire life in all of your cells and is never used for anything. That's the idea of a recessive allele. If you've got 2 recessive alleles, then your cells don't have any choice except to use that recessive allele as the gene that's going to be used to make a protein. And another thing to think about is that all of our cells have the same DNA. We started out with the DNA that we got from our mother and our father. That cell divided. The 2 cells produced have the same DNA, and every time cells divide, that same DNA is passed on to the new cells that are produced. Well, in terms of gene expression, all those genes that are in our DNA, those 25,000 genes, if we have that many, they're not all expressed all the time. Some of them are expressed while some of them sit dormant. That's what we're going to look at. Some of them are expressed while other ones are never expressed. Some of them are expressed when the environment changes, when there's different sources of food, when there's different temperatures, when there's different salinities. Those environmental conditions can cue some gene to turn on, and you start making that protein. But the other times, that gene is just sitting there not doing anything. Another thing is that this will be evident when we look at this, it'll be obvious, is that some genes are turned on during some time of development and then they're turned off. Some of them are turned on early on and never again your entire life. And then finally, this is the example with the nerve cell and a skin cell, that they've got the same DNA, but they're obviously very different from one another because certain genes are expressed in the skin cell that aren't expressed in the nerve cell. And certain genes from the nerve cell are expressed that aren't expressed in the skin cell. And there's other ones in each one of them that are turned off. They're never expressed. So let's look at this. This is this dominant recessive allele idea that I mentioned before. Remember, we've got 2 alleles for a gene, because we've got 2 parents and we've got 2 chromosome number 1s. We've got 2 chromosome number 2s. We've got 2 chromosome number 3s. Chromosome number 1 has gene number whatever, and so does the other chromosome number 1. In some cases, those genes are the same. The expression of those genes results in manufacture of the same protein. That's homozygous. In other cases, the genes are different. The gene that you got from your mother on chromosome number 1 is different than the gene you got from your father on chromosome number 1. And one of those is going to be expressed and the other one isn't in a lot of cases. So just the fact that there's dominant alleles and there's recessive alleles is a form of gene regulation, where the dominant allele is expressed and the recessive one is never expressed. It's never used to make a protein. That's a form of gene regulation. There's a lot of environmental conditions that cells go through or organisms go through that result in certain genes being turned on. Here's an example with bacteria that's pretty simple, that under some conditions, if there's no glucose, for instance, then the genes of bacteria can be turned on and they start to synthesize this enzyme that can be used to break down other energy sources besides glucose. If there's no lactose there, another type of sugar, then there's another gene that makes another enzyme that normally it would be expressed and that enzyme would be produced and it would be used to break down the sugar lactose. But there's no lactose. The bacteria senses that and it stops making this protein. There's no point in making this protein because there's no lactose for it to digest. And then when it experiences lactose or it comes in contact with lactose, it starts expressing that gene, starts making that protein that can digest lactose. Same thing with glucose. So it depends on the environment. When there's glucose absent, stop making that glucose digesting enzyme. There's no point. It's a waste. When there's lactose absent, stop making that lactose digesting gene because it's a waste of energy, it's a waste of resources, there's nothing for it to do. So that's an example of the environment. And as I said before, it could be temperature. When it's a certain temperature, there's certain genes that are expressed and other temperatures not. Oxygen could have an influence on gene expression. pH, a number of different environmental factors turn on some genes and turn off other ones. Here's another thing that we've all experienced, we're all a little familiar with anyway, is this idea of development. If you looked at what we looked like during our development, at some stages in our development, we've got a tail. Some stages of our development, we have webbed digits, fingers and toes. When we're born, we don't have a tail. When we're born, well, some people have, but it's not very common. When we're born, we don't have webbed fingers, although, again, some people are born and they have to have their fingers operated on. And we have gill slits during a certain phase of our development. When we're born, we don't have gill slits. So during our development, we've got genes that are turned on that result in this tail being formed. And then those genes are turned off, and then we lose our tail. We've got genes that are turned on that result in the formation of these gill slits. Then those genes are turned off, or other genes are turned on, and those gill slits disappear, and we're born without gill slits. Same thing with the webbed fingers and toes. It's present during some of our development, but not others. So as you recognize, during that 9 months of pregnancy, when we're all little babies developing in our mother's womb, there's a whole bunch of genes that are turned on as we're going through these different developmental stages that are never turned on again our entire lives. And there's a lot of genes like this. I'm a lot older than most of you guys, and I've got genes in my body that aren't being expressed anymore that were when I was younger, when I was your age. And there's genes being expressed in me because I'm older that weren't expressed when I was younger and aren't being expressed in you. So as we move through developmental stages, some genes are turned on and some genes are turned off. Well, here's an example that have used before. You've got Michael Jackson and Michael Phelps, these two Michaels. You know, if you looked at what they looked like when they were younger, when they were 15 years old, Michael Phelps looks a lot like he looks like a 15-year-old Michael Phelps, just younger. Michael Jackson looks like a completely different person. You know, very different when he's this age versus 15. But they both got the same DNA, and that DNA is expressed. Who knows what was going on with Michael Jackson to make his appearance change so drastically. Anyway, let's move on to this last point that I wanted to make, is that if you look at cells, cells have the same DNA. So here's an example. You know, this is this nerve cell or an epithelial cell, which is a skin cell, this example that I used. These two cells do very different things. They have very different functions, different shapes, different structures. But if you look at their DNA, this is one of our cells. This cell has the same DNA as this cell. So why are they so different? They all started out from a single fertilized egg years ago, and they've gone through mitosis, changing, and then eventually you've got, here's this nerve cell where this little green part, that's part of the DNA that's expressed that results in traits that this neuron has. If you look over here in the skin cell, this green part, this gene is not expressed in a skin cell, so that skin cell doesn't look like a nerve cell. The nerve cell looks like a nerve cell. If you look at this part of the DNA, this part is expressed. It results in synthesis of particular proteins that contribute towards this epithelial cell looking the way that it does. These genes aren't being expressed in the neuron. So these two cells have the same DNA. They just have different parts of the DNA being expressed, different genes being expressed that result in different types of cells that allow them to carry out different functions. Okay, well, there's a whole science of sometimes looking at problems that happen and sometimes looking at trying to come up with solutions to genetic problems. But there's all kinds of opportunities for cells to regulate genes being expressed to make a protein. In bacteria, it's not that complex. You've got DNA that's used to make RNA that's used to make proteins. In eukaryotes, like our cells, the DNA is in the nucleus. And so you make RNA based on the information in DNA. And we saw before some examples of that RNA being spliced and diced and modified, some of it cut out and some of it. So there's some messenger RNA that's made that doesn't leave the nucleus. It doesn't go to the ribosomes. This gene can be expressed and you can make messenger RNA, and that's where it stops. So it's at the site of transcription. You can have messenger RNA made and go off to the ribosomes. And so this is post-transcription. But when it gets to the ribosomes, there's some kind of something is wrong or something is missing, or there's some signal that's not there. So it doesn't start being conveyed towards the assembly of a chain of amino acids. So this messenger RNA might make it to the ribosome, but it doesn't result in the synthesis of this protein. This blue strand coming off here is a protein. Another thing is that this messenger RNA could go out to the ribosomes, and it matches up with these ribosomes, and we get the ribosomes moving along. But for whatever reason, then this chain of amino acids isn't made. That's translation. Translation doesn't take place. Or you've got some where this blue chain of amino acids is formed, but it doesn't form bonds with one another. It doesn't take on its three-dimensional configuration. So you've got a polypeptide chain, but the events that are necessary for that polypeptide chain to turn into a fully functioning protein don't happen, in which case you've got post-translational regulation. And cells can regulate the expression of genes. It's not just that there's no going back once you make this messenger RNA. You could make messenger RNA, so in a way that gene's been expressed to the point where you've made messenger RNA, but you've still got a long ways to go to a fully functioning protein, and that could be regulated during transcription. It could be related post-transcription. It could be regulated post-translation even. There's a lot of opportunities for eukaryotic cells especially to regulate the synthesis of proteins. There's a lot of control. Proteins are necessary. It's not surprising that there's so much control over the regulation of proteins. So that's a very short summary survey of looking at gene expression. Now we've looked at a lot of different things, and we're going to be ready to move on to a lot of different aspects of what can go wrong when we look at genes and how we can use this information to make a better world for humans, especially in terms of health.